Good day, everyone. I'm Ivan, the moderator for this webinar. I welcome everyone for today's webinar, which is GMP Environmental Monitoring for Pharmaceutical Clean Rooms 2024. I would now like to introduce our presenter for today, Roger Cowen. Roger Cowen is the founder and owner of our Cowen Consulting Services LLC. He has 35 years of experience in pharmaceutical quality assurance and manufacturing. Roger's area of expertise includes aseptic pharmaceutical manufacturer, GMP facility audits, technical transfer, microbiology, environmental monitoring, contamination control, product sterilization, process development, process validation, clinical supply manufacturing, labeling and distribution, US international regulatory requirements, regulatory submissions, and quality assurance and control. I would now like to hand over the floor to the speaker. Over to you, Roger.
Thank you very much. So my name is Roger Cowen, our Cowen Consulting Services, and I'm here today to present a webinar on the GMP environmental monitoring for pharmaceutical clean rooms. Clean room is a room in which the air supply, the distribution and filtration, materials of construction, operating procedures, and personnel are regulated to control airborne particle concentrations so that the appropriate air cleanliness levels can be met. Environmental monitoring and environmental control. Environmental control refers to the various systems employed to ensure that clean rooms operate within the prescribed parameters. In contrast, environmental monitoring, that term looks at the end results of the environmental control system. In other words, the micro and particulate quality of that clean room. So let's look at a few of the GMPs that apply to this. The FDA Guideline on Aseptic Processing came out in 2004. Now, another couple of GMP references here, 21 CFR 211.42, another GMP reference, 21 CFR 211.113. So those GMPs discuss the need for an environmental monitoring program. Here are some of the benefits of such a program. Now, when you set up an environmental monitoring program, make sure that your SOPs are very comprehensive. And I've got a quick list here of what needs to be included in those SOPs. Now, how about the testing requirements for the GMP? First off, microbial testing. Now, the other major testing that you'll be performing is non-viable particulate testing. Now, other parameters that are tested as part of the environmental monitoring program is temperature, humidity, and differential pressure. So let's move on to action and alert levels for environmental monitoring. You can set your action levels based on the published regulatory specifications that we just saw, particularly with the non-viable particulate monitoring systems. Obviously, routine measurement is needed. Okay, so how do you measure non-viable particulate? Most common technology used is the laser optical particle counter. So many companies have adopted a continuous, automated, real-time remote system for these particulate monitoring systems. Now, the probe should be sampling air at about working height. Now, switching from non-viable particulate to microbial monitoring systems for air, there basically are two types of air monitoring systems. It's called passive monitoring and active sampling. Now, switching from passive to active air sampling, and one good example of an impaction device is the STA sampler, which stands for slit to air. You can see a photograph to the right. Another impaction device is a sieve impaction sampler. So there's another type of active air sampling, and that's using filtration devices. So the third type of device that's used these days for active air sampling, and it's a recent innovation. It's a real-time counter. It uses an ultraviolet laser. Okay, so we're going to switch from air to surface. So there are two methods for sampling surfaces for microbial contamination. There's contact plates. Okay, another surface monitoring system for microbes are swabs. Okay, so what happens when you're microbially monitoring personnel? Now, personnel are actually the biggest source of microbial contamination in your clean room. Now, when you're monitoring these personnel, you can use the glove print. In terms of sampling the garments, going back to the lab here, it's very important that all this media that's used for sampling has controls on it. How do you choose your sampling location for microbial? How about frequency? Well, for grade A and B aseptic areas, You should perform microbiological sampling at the end of every production day or shift. Now, for grade C or D clean rooms, you can perform microbiological sampling on a weekly, biweekly, or monthly basis. Now, how about the sterile isolator or RABS technology clean rooms? Okay, so investigation of alert and action level excursions. Microbial identification. Okay, how does this all relate to release of product? The release does require a review of the environmental data. Now, documentation. A summary of environmental data from the batch is usually included in that batch record. The environmental monitoring data should be summarized in trend reports. Now, this data is summarized in a periodic report prepared by QA, presented to manufacturing, engineering, general management. Now, there's been a new measurement that's been introduced by the USP. This is called percent contamination recovery rates. So the USP has a table in it. Here's a few considerations for graphing. Here's some examples that I've put together. And here's another one that compares months of sampling. This one here is the percent recovery rate. This is what we were talking about with the USP. So that brings me to the end of our presentation.
Yes. Okay. So first off, thanks very much for listening. I realized there was a heck of a lot of detail in this, but I think that's what you want because this is a very involved subject. So again, thanks for that. I hope there's some little nugget of information that's in this webinar that you can use in your own facility. So thanks for listening.
Thank you.
Thank you, Roger.
Okay, thank you. Bye.
That concludes the session for today. Thank you everyone for participating. You may all disconnect now. You have a great day ahead.